and will be turned off when nutrient is no longer needed. When excess arginine is present, it binds to the repressor and changes its shapes. Then the repressor binds to the operator and blocks the arginine synthesis. So another repressible operon is the tryptophan operon. The tryptophan operon encodes genes for the biosynthesis of a amino acid tryptophan. The operon is not expressed when the cell contains sufficient amounts of tryptophan. The operon is expressed when the levels of tryptophan they are low. The tryptophan operon is negatively regulated by the tryptophan receptor proteins. So this repressor binds to the operator to block the transcription. Binding of repressor to the operator requires a co-repressor, which is tryptophan. Low level of tryptophan prevent the repressor from binding to the operators. So this is the situation. Let us say this is the inactive repressor at the moment, tryptophan repressor. Tryptophan is not sufficient. So this repressor is there in the inactive form, and this is the operator which is going to transcribe all of the genes which are required, A, B, C, D and E. All of these are the enzymes which are being switched on to make the amino acid tryptophan. Once the tryptophan is more than required, so this excess of tryptophan is going to bind here on this repressor. As a result of this binding, so its shape become complementary to bind with the operator. It binds here with the operator, and this RNA cannot proceed further. RNA is blocked to transcribe these gene E, D, C, A, B, A. So these genes are, and then ultimately there is decrease in the level of tryptophan. In bacterium E. coli, a group of genes that code for enzymes required to synthesize the amino acid tryptophan, all 5 genes are transcribed together. So they are going to make a group called as operons. So these are these genes which are there controlling the synthesis of amino acid tryptophan. This is the area of promoter in which this is the operator, and this is one of the repressive protein which is going to repress or blocks. If the low level of tryptophan is there, then this mRNA is allowed to transcribe all of these 5 genes at the same time, and the tryptophan is being made by using you can say the ribosomes. But if the tryptophan is more than required quantity, excess of tryptophan, it binds with the repressor protein, and once it binds with the repressor protein, so it is going to sit at the operator, and this messenger RNA is going to touch here, and that is not allowed to transcribe. So what is actually happening? This is the repressor protein made up of polypeptide chain having helical forms, and these helical forms they normally cannot bind to this you can say the DNA molecule. But once these excess of the tryptophan is going to combine there, so they are going to combine here, and once it now its shape is like that it can chloramphenicol, linomycin and tetramycin binds to the ribosome and block elongation. Streptomycin is going to inhibit peptide initiation and elongation. Controlling gene expression is often that is accomplished by controlling the transcription initiation. So the regulatory proteins, they are going to bind to the DNA either to block or to stimulate transcription depending upon how they interact with RNA polymerase. So this is very, very important that the regulatory proteins must be there in the activated form. Prokaryotic organisms regulate gene expression in response to their environment, while the eukaryotic cell regulate gene expression to maintain homeostasis in the organisms. Dear students, so we have learned a very basic mechanism that around each cell there is a tissue fluid, and the composition of tissue fluid is maintained within a very narrow range. If the amount of glucose is less, so there are certain chemical processes, it is increased. If the amount of glucose in this environment is more, so if anything is increased, there are certain processes which are going to decrease it, and if anything is going to decrease, then that element, that compound is increased. This is called as homeostasis, maintenance of constant and required to use that new product. So this is a, you can say, the beautiful control mechanism given by the God to regulate our machinery at the cellular levels. Continuing our slideshow, controlling the expression of eukaryotic genes require transcription factors. Journal transcription factors are required for transcription initiation. They are required for proper binding of RNA. Polyphasic transcription factors increases transcription in certain cells or in response to the signals. Looking in this animation, control of gene expression in eukaryotic cell, that is slightly different in prokaryotic cell, because most of the prokaryotes you can say they are having both the processes occurring in the cytoplasm. But in case of eukaryotes, so DNA is there in the nucleus and there are activator proteins which are going to make the copy of DNA, and this is the enhancement of this process. mRNA is going to come making the multiple copy of this transcript of RNA, and then this RNA is given the 3' and 5' end. So post-translational modification is there, there is cutting of the intron and exons, and then you can say all of these activated. This is the mature RNA for calcitonin, and this is the mature RNA for calcitonin related proteins. And again you can say once whole of this process is going to pass through this pore. This is the nuclear pores which are present in the nuclear membrane. So this is going plasm. So in the cytoplasm, it involves the control of transcription initiation, but